Dear students, I hope you all will be fine. I'm here, Muhammad Jamal Khan, lecturer in botany, University of Education, Lahore. Today, we will discuss a topic from one of the core subjects of botany, that is plant systematic anatomy and development. The course code of this subject is BOTN 1112. So let's begin this lecture number 17, and that is related to one of the major component of plant cell, that is cell wall. So here we have a general structure of plant cell. As you see, there are a number of organelles and suborganelles that is present in the cell. You already study about this plant cell structure in your previous classes. So over focus in this slide, here we have a cell wall. So as you know, when we give a very brief definition of the cell wall, we can say that cell wall is the outermost layer of a plant cell, and it is the only non-living part of the cell that is secreted by the protoplasm for its protection. Its formulation, as you know, take place at telophase of the cell division. So here we have one of some of the features of cell wall that reflects its importance in plant cell. As you know, in young cells of plant cell, the cell wall is thin and delicate, but in a large cell it becomes thick and strong. In addition to this, as you know, this is the major component of a plant cell, and it is a non-protoplasmic compound or subject. Due to this, because it is the byproduct of metabolic activity of protoplasm. The major role of cell wall is to provide support and protection to the cell, and it also play a very important role in adsorption and translocation of various substances and secretion of different products in response of metabolic activities. The morphology and the composition of cell wall vary up to a different extent. So basically, cell wall consists of 3 layers. One is middle lamella, the second one is primary wall, and the third one is a secondary wall. So here we have another pictorial view of cell wall. As you see here, this is a cell wall, and basically cell wall consists of 3 layers, but firstly we describe 2 layers. The first one is middle lamella, this one, and the second one is the primary cell wall, and the third one is secondary cell wall that we'll discuss later in this lecture. So first of all, here we have the cell wall. This indicates the cell wall, and this is also indicate the cell wall. So here we have 2 layers. The first one is middle lamella, and the second one, this one is primary cell wall. So a middle lamella is just nothing. It is a layer between two cells. Suppose here this one is the cell wall of one cell, and this one is the cell wall of second. So here I give the one, this is our cell number 1, and here this is the cell number 2. So in between these two layers, in between cell number 1 and cell number 2, here we have a black or gray shaded layer, and that layer is called middle lamella. So it is a layer between two cells, and it helps to attach cells to each other. And majorly this middle lamella or gray shaded structure is composed of calcium pectate. After that, here we have a primary cell wall. This one is a primary cell wall, and here this one is a primary cell wall. So in between these two primary cell walls, there is a presence of middle lamella. So now we discuss about the primary cell wall. I give you a very brief account of primary cell wall. Basically, a primary cell wall found around a young plant cell on the outer side of plasma membrane. The primary wall is very thin, elastic, and capable of great extension. It consists of cellulose, hemicellulose, pectin, and sugar compounds. So now we discuss about the characteristics of middle lamella. As you know, each cell has its own cell wall in case of plant cell. The cell wall of adjacent cell is joined by a specific layer that we call as middle lamella. Middle lamella is a thin layer, and cellulose is absent in it. It is mainly composed of a specific substance, that is pectin. Middle lamella can easily be dissolved by applying certain chemicals. So by using these chemicals, it separates the cell. So when we study about the cell, then we should remove this middle lamella. The removal of middle lamella is called maceration. In addition to this, the middle lamella is considered to be the intercellular substance because it holds the cell together. Here we have the characteristics of a primary wall. Primary wall is flexible in nature and permeable also. Due to this, the primary wall is elastic and transparent. Number of material passes through it. It is the first wall formed in the developing cell, and it remains primary in many types of mature cell. It is only present in living cells only, and it is absent in non-living cells like tracheids and sclerenchyma. In living cell, we give the example of meristem cells. Primary cell undergoes extensive growth that result in increase in size of the cell. It is chiefly formed of cellulose and hemicellulose and pectin material as far as concerned its composition. The increase of the thickness of the primary layer occurs due to the deposition of successive layers of cellulose towards the inner side of the cell. In addition to this, lignin is deposited between the primary wall and the middle lamella in mature tissues. Therefore, the primary wall cannot be differentiated from the middle lamella due to deposition of lignin substance. The primary wall of many cells has lamella-like structures that we call as microfibrils. These microfibrils play a very vital role in the stability of the primary wall and in the maintenance of shape of cell. And these microfibrils are arranged in a crisscross pattern. Due to this, a cell can maintain its shape. So here we have different chemical constituents that arrange in a specific pattern to form a primary wall, including middle lamella and primary wall. These chemical constituents actually are the major component of cell wall. As you see here in yellow thread-like structure, these yellow thread-like structure that indicate a substance or chemical constituent that is pectin. In addition to this, here we have a green color strands. These green color strands that indicates a subchemical constituent that is hemicellulose. A blue colored dots here, here, and here. These blue colored dots indicate a specific substance that are soluble proteins, and these rod shaped structure that indicate in sky blue color, these indicates cellulose microfibrils. This upper layer or joining layer that join two cells together, or cemented layer, that is called as middle lamella, and below this middle lamella we have a primary cell wall, and different chemical constituents like cellulose microfibrils, pectin, hemicellulose, and soluble proteins arrange itself to form a very compact structure that we call as cell wall. Now we discuss the third layer of cell wall, that is secondary cell wall. As the increase of surface area of a primary wall stops with the stoppage of growth of cell, now the secondary wall is formed inner to the primary wall. One important thing, that is the secondary walls have no surface growth. Only increase in thickness occurs in it. This secondary wall is composed of a pure substance, that is cellulose. By nature, the secondary wall is tough and strong. Sometimes it grows so much that it fills most of the part of a hollow cavity, that is lumen of the cell. So due to the growth of the secondary wall, as I already told you, it covers the most of the cavity of lumen, like tracheids. So in tracheids and fibers, the secondary wall composed of three layers. The outer layer is S1 layer, then we have a middle layer, that is S2, and then we have a inner layer, that is S3. And some cells have more than 3 layers. In these layers, the layer, the middle layer or S2 layer is the thickest in all of these three layers. Later we will have a slide in which it clearly reveals the thickness of middle layer or S2 layer. And this secondary wall is mainly increased due to the deposition of lignin. So the deposition of phenomena in which there is a deposition of lignin, we call it as lignification. So in this slide we have a very good demonstration regarding a cell wall. So here we have the outermost layer that of cell wall, that is middle lamella. So this is the cemented layer between two cells, or this is the layer that join two cells together. So this is the middle lamella. Then we have this wall, that is a primary wall, and after that, after primary wall, toward inside we have a secondary wall. Here you clearly observe it here. So the secondary wall, the thickness of secondary wall, as I already told you, is thus due to the deposition of lignin. So this deposition of lignin increase the thickness of secondary wall. Then we have proteins in secondary wall, we have the semicellulose substances, and we have the cellulose. So actually cellulose, hemicellulose, and lignin and protein are the chemical constituents that made the secondary cell wall. So here you clearly observe the thickness of secondary cell wall. In addition to this, we discussed that in secondary cell wall we have three layers in tracheids and fibers. So these three layers of secondary wall are indicated as S1, S2, and S3. Among these three layers, the thickness of S2 or secondary wall is greater than S1 layer and S3 layer. So here you clearly observe the difference, and the thickness of S2 layer, this thickness is greater than S1 thickness and S3 thickness. And sometimes due to the unequal distribution of lignin, or due to the unequal lignification, it produces different thickening in secondary wall. So due to the unequal lignification or unequal deposition of lignin, we have different thickenings in secondary wall. So here we have, first one is annular. These are ring-like thickenings, and in this case a ring of lignin are deposited one above the other. The second one is scleriform. These are ladder-like lignin thickenings. And the third one is spiral. In this case thickenings are arranged in spiral bands. So here we have a pictorial demonstration. So these are ring-like thickenings. Here in case of annular lignification, these are the ring-like thickenings, as you observe clearly in this. These are the rings. Then we have a spiral thickenings. This one is the spiral. Here, this one is the spiral. And here we have a spiral thickenings of lignifications. And the last one, and the third one is scleriform. So here we have scleriforms. Next, we have a secondary thickening, that is reticulate. In this case, lignin thickenings form a network. The last one is pitted. In the secondary thickening, referred to as pitted, the entire cell wall is thickened and it has only few unthickened areas called pits. And if these are simple pits, then we call it as simple pits. And if these thickenings or unequal unthickened areas have bordered or thickened around itself or thickened part around itself, then we call it as bordered pitted. Here I have the brief summary of this lecture in a very simple slide. So first of all, we discuss a cell wall. Cell wall consists of 3 layers. As you know, the middle one layer is the layer that cemented 2 cells to each other, that is middle lamella. So here we have the middle lamella, this one is the middle lamella. Then the outermost part of the cell, that is primary wall. So this indicates the primary wall here and here. And then inner to the primary wall we have a secondary wall. Here we have the secondary walls, and the secondary wall on the base of their thickening divided into 3 further layers, and the layers indicated as S1, S2 and S3. In between these 3 secondary wall layer, the S2 is more thicker than S1 and S3, and sometime due to the growth of this secondary wall, a cavity that is present hollow is also covered by a secondary wall as in the case of tracheids. This is all about for this lecture. Thank you.